think you're connected, right? Or...
So it says connected, but let's see. I'm here. Success! Okay.
Cool.
Actually, this worked. I don't know if Sime still wants to talk to me now. We'll see.
I can slack her.
Yeah. Cool. I'm glad I got this working. Okay, and now I can plug in the mic. Hello, hello. We are back. We now have working internet. Yay! Let me announce on Twitter that I am now connected.
Just gotta finish this up.
Okay. All right. All right. Hello. So we're now ready to talk ticks. I'm pretty excited. Let's see. Hopefully now we can have a few people join in when they can. Cool. Let's see here. So I think we have... So we have just me here for now. Okay. The next meeting starts in 10 minutes. So, let's see. What should we do? Maybe I can go talk to Sime briefly, and then I'll come back and talk to you. And we're gonna head over to the Bigwigs lab, Bigwigs location. And hear a little bit about ticks and why they matter. Opening doors is hard with this. Wait. All right. Hello.
Hey.
I'm back. I know this. We have to go soon, but do you think you could briefly talk a little bit?
Sure. I was literally about to start a phone call though.
Oh, okay.
Like I have like 1 minute.
Yeah. Go for it.
Okay.
Yeah. All right. So. Oh, it's all right. So.
I either have 1 minute for you or.
Oh, I misunderstood. I thought you mean
Yeah.
you wanted to start your.
Otherwise, because I'm like texting with someone. We're about to start a call.
Yeah.
Maybe we could do it next week.
We can do it next week. I can I can just talk. Yeah, I can just talk with Fauna and them. Okay, sounds good. All right. Busy PIs are always busy, so we're gonna keep on listening. Keep on finding some cool people to talk to. So we're here. Hello.
Hi.
How are you?
Good, how are you?
Hello, everyone. This is Fauna.
Hi.
Fauna is a graduate student in Sime Chow's lab. Did you want to show us some cool stuff?
I can show you our ticks, our tickery.
Or is that what you call it, your tickery?
That's what I've heard it called.
That's funny.
I
I like that.
We call this the tick room.
The tick room.
They are supposed to be handled in here.
Ah, okay. Let me can I pull up some let me pull up the ChewLab website
Oh yeah.
and just have that be ready. Cool. Yep. All right. And we're good. Cool. So we have some cool ticks to show you here. So do you have a few different species here, or what's going on? How can you tell that these are different species?
So we do have different species. It takes a little while, but eventually you get to know them by looking at them. So the Ixodes species are, in North America, Ixodes scapularis can transmit the Lyme disease bacterium.
Yeah.
So that's what we primarily work with here. And I don't know if everyone can see, but the females are a little bit larger than males, and they
Mhm.
have like a round bottom or a round abdomen, or a red abdomen
Mhm.
and round.
Yeah.
Um,
Maybe you can zoom in a little bit here on the camera.
I think they
Hopefully
can
it'll
see.
come up.
There are nice pictures available on Google or our lab website, actually.
Yeah, maybe we can find some of those.
Yeah, I think if you go to research, I think
Yeah.
the header should be a nice tick photo. Yeah. So
Oh, cool. So what species is that?
that's the female Ixodes scapularis tick. So very prevalent on the East Coast. There's many campaigns to look out for these ticks because if you get bitten, you're at risk for getting Lyme disease. And then the males are very similar, but they're a little bit smaller and they're largely all black, so they don't have this bright red abdomen.
What's the red color from?
I presume it's a pigment, but I don't actually know. And since we don't really have good genetic information, I'm not sure we know exactly what's like contributing to this red color. But maybe we can find out soon one day.
Yeah, and is the so it's a pigment. It's not from the heat. It's not from feeding.
Oh no. We actually don't have any fed ticks, but when they feed, they get rather large, so they'll look like a little bean, and they lose this red color, so they actually turn into like a gray color all over. So they look pretty different when they fed.
Huh, I wouldn't have expected that. But let me adjust my screen so I can actually see the questions people have.
Okay. And we do have other ticks.
Cool.
So we unfortunately had a mass die-off of our ticks.
Oh no.
We're not sure what happened, but we do have quite a few species.
Mm-hmm.
So these are the Lone Star tick, Amblyomma americanum. And you can see they have a little white, the females have a little white dot on their back, which is quite appropriate given their name.
Yeah.
And these ticks, if you get bitten by them, you can potentially be exposed to a couple tick-borne pathogens, but these have been in the news as possibly contributing to non-mammalian meat allergy. So if you get bitten by these ticks, it's possible that you'll no longer be able to eat beef or pork or, typical mammalian meat that's in American diets.
Oh wow. Do you know why that is? I think I heard about that. Sorry, I'm just gonna add our video here so we can tell if we can actually show our our image. Whoops. If we're actually showing people what they can see. Cool. Cool. Here we go. So.
Okay. And
So with this, the meat allergy, I've heard it has to do with is that alpha galactase?
yeah, so alpha gal is not only, from what I've read, it's not only found in the Lone Star tick saliva, it's also found in other tick species saliva. But they think that it's a modification found on like tick proteins and also in meat that's not in. We lost that modification many years ago, or a long time ago in evolution,
Mm-hmm.
and so that's why people think we're mounting an immune response when alpha gal is introduced to us via a tick bite or in your diet. So I don't know too much about non-primate meat allergy, but that's basically my understanding. But I did want to show, so I think one of the very cool and interesting things about ticks is how different their life stages are. So the ticks we work with have 3 different life stages, and I don't have the larval stage. but I can tell you they're about the size of a pin. They are very, very small. And then when we look at amblyomma ticks, they will molt, and so then they'll molt to be about the size of a sesame seed. I don't know.
Oh wow, that's really small.
Yeah, they are. They are very small, but at least you can see them at this life stage. It'd be very hard to see the larvae.
So for the larvae, you have to be under the microscope to see them?
You can see them by eye, but it's difficult to focus. They are very small.
Mm-hmm.
And then,
but like with like a 10x magnification, it's fine, or?
Oh yeah, I think you could see them fairly well. I don't look at the larvae that often. I mostly just move them from tube to tube. I don't like get up close and personal with them.
Got it.
But then, so the larvae will molt into the sesame seed size tick, which I hope is
I think
showing up. And then after this nymphal stage tick feeds, they can molt into these rather large adults. So this is the female, which is...
Yeah, the feedback's a little laggy, sorry.
Oh, okay. So the female is pretty large. I'd say, I don't know, maybe at most a centimeter in length in terms of body size. So you can definitely see these coming if you're out and about in nature.
So how are these sex separated from the beginning? When do you separate them? Like when does that process happen?
So as of now, we don't have a way to distinguish males and females at the larval or nymphal stage. There's no like visible marker that we know of, and as far as I know, there's not a genetic marker as well. But when they get to adulthood, for Amblyomma americanum, the Lone Star tick we're looking at now, and also Ixodes scapularis, you can tell just by looking at them that there are males and females. Usually, the females are a little bit more ornate, so these have the Lone Star on their back, which is like a little bit shiny. And then with Ixodes scapularis, the females are red, and the males are a little bit smaller and nondescript.
That's kind of the opposite of like birds, right? You think of like male ducks are usually the fancy looking ones, or peacocks, right? And
Yeah.
the female birds are usually the like lame looking ones.
Yeah, it is. I'm not sure why that is, but that's just the way ticks have made things work.
Yeah. Yeah, cool.
Yeah.
Cool.
So these are our ticks. They just hang out. Oh, do you want to show the incubator?
Yeah, let's look at the incubator.
So they just hang out in this incubator. We keep it at like room temperature essentially, and we have a light that is on. I think it's an 18.8. Sorry,
18.6.
18.6.
Yeah.
Or maybe it's 16.8.
That would make sense.
So it's basically trying to trick the ticks into thinking that it's summertime all the time, so that they're active, and they will be ready for us if we want to do experiments with them. And
What if you wanted it to be in winter time? What kind of light cycle would you do then?
probably more like a 12-12, and we'd also, I mean, depending on the experiment, I might put them at a colder temperature. Because ticks do survive outdoors, and so they can survive at colder temperatures. They're ready for it.
Oh, cool. Yeah, obviously they've been out there. So
Yeah.
why do you want them to be do you want them to be thinking it's summertime?
I think we found that our ticks will survive a little bit better if they think it's summertime. Also, I know that there was a study or a paper published a while ago about how to keep ticks in the laboratory, and they suggested the light cycle that we use. I think they tested a number of different light cycles and temperatures, and they found that in general the ticks seem to do a little bit better in captivity with these settings or in this situation.
Are there any long-term effects, like if they think it's summer for 3 years, does that affect them at all?
It likely does. I haven't had the chance to read this paper, but I know recently a paper was published where they talk about the long-term effects of starvation on ticks and how I think it speeds up their metabolism. And so, yeah, it's likely doing stuff that we don't fully understand, but we're working on it.
Cool.
Yeah.
Very cool.
I'm going to put these back.
Yeah. So what are the different containers you have here?
So we have other species, which are basically non-Ixodes species.
Mm-hmm.
Sometimes we use these for like one-off experiments or if we're trying to compare between tick species, but they're generally not the focus of our lab at the moment. We have Ixodes species, so Ixodes scapularis on the east coast, and on the west coast we have Ixodes pacificus, which is closely related and can also transmit the Lyme disease bacterium. And then we have a jar of ticks that have been fed and maybe molting. So you can see, so these are Ixodes scapularis nymphs that have been fed.
Whoa, they're tiny.
They are tiny, and these are larger than they were before they fed. There may be like a swollen sesame seed or like a small apple seed, I would guess. And so
I like your use of seeds as the sizing difference. That's nice.
I think that's what I've seen, so it's easy to work from that. And so these are just going to hang out for about 4 to 6 weeks until they molt, and the lighting condition of summertime also helps promote their molting, because if they thought it was wintertime given the lights, they might try to wait until winter was over before they emerged into the new life stage. So that's one more benefit of the light system. So yeah, these are just going to hang out in a tube until they hopefully molt. They don't always, but yeah,
Just life happens. Yeah.
it does happen. And then we have some practice ticks that maybe have survived some experiments and could be used to like teach someone how to dissect a tick, or how to inject into a tick, because these are all things that take practice.
Wow, that's kind of funny. You have practice ticks. Does that just mean they're like not as carefully taken care of, or what does that mean?
It could mean they're moldy. It could mean they were provided to us by someone outside the lab. So we get tick donations often, where people will bring us ticks that they found. It could mean that maybe I was practicing injections on a tick, and I wanted, like, there's nothing wrong with it, but maybe it's not the best species to use in an experiment, or like it's not the best subject to use in an experiment, and so I'll just put it aside so that maybe I can help someone else learn with this tick. So that's our practice tick.
Yeah. Well, we have a question from Zultra Lord about whether ticks could ever be used as a delivery mechanism for weaponized pathogens.
I would speculate that that's certainly possible. I don't know about any time in the near future. I feel like we don't even understand the existing pathogens that are transmitted by ticks. So manipulating those pathogens and making sure that a tick would still carry them and pass them on, I think would be tough. So I wouldn't lose sleep over it, but it's not the craziest thing.
But it could make for some good sci-fi.
Absolutely.
Yeah.
Yeah.
Yeah, because Zultra Lord pointing out that mosquitoes could always be the next plague source.
It's true. Yeah.
But yeah, there's a lot of vectors that could be working on. Yeah, let's go in there. That could be the next bad vector.
It's true. And vector-borne disease is on the rise.
Is that true? Vector-borne diseases are on the rise? Do you think that's a reporting issue? Or do you think that's availability? We're going back into the lab now.
I think it's a little bit of both. I'm sure there's more accurate reporting or diagnostic tools, but I know that climate change is also thought to be playing a role.
Mm-hmm.
So ticks have longer seasons to breed. They have more of the year is warm, and so not only are we out and active, but so are the ticks, and so it's more likely that you'll encounter them, and then that puts you at risk for getting a tick-borne pathogen or being exposed to one.
So even just like a couple more days in the summer or warm days, that can affect the number of incidences of tick-borne pathogens?
I've seen that as being attributed to the rise that we're seeing.
Oh, interesting. Cool. So, and you had some questions for me about the RNA-seq data. Do you want to talk about that?
Oh yeah, I would love to.
Sure.
So I am currently doing an RNA-seq experiment where I'm trying to look at hemocytes, which you can think of as like white blood cells in the tick. And I would like to know what are your thoughts on quality control when you're doing an RNA-seq experiment, because I've been told that it doesn't seem to make too big of a difference if your data set is large enough, but it would be great to hear your input.
Yeah, sure. I've worked with RNA-seq data sets that are both quality controlled and not, and largely the results are the same. Like the top differentially expressed genes are largely the same. If you want to be super careful, that's I think always appreciated. You can trim the adapters. You can trim sequences of reads below a particular quality control that are like all bad in a row versus like kind of like a checkerboard. Some of them are bad, but like if they're all bad in a row, that's gonna be bad. And there's a lot of tools for that, like Trimmomatic, Cutadapt, there's a bunch of them.
Okay.
I have found that like largely results are the same, and most aligners nowadays are pretty good to know that this if your read does have part of an adapter, it's really only going to match the part of the read that matches the genome.
Okay.
So if like say the last 10% of it is an adapter, then it'll only match the first 90%, and it'll what's called soft clip the last 10%, and it will say that hey, this last 10% was part of the read, but it didn't match in the genome. So the downstream programs that end up counting genes won't count those bases that are soft clipped.
Okay, that makes sense.
So I can see people being like, oh, you have to do it, but also people being like, yeah, it doesn't like super matter, because in the like global analysis, I found that I can't say it doesn't make a difference. Like it certainly like makes some kind of difference in subtle changes, but I think overall it's not as important as having a good experimental setup and having a good controls, like that you're like you have enough replicates, that your experiment was like cleanly done, there's no contamination. I think that's way more important than the trimming and stuff at the bioinformatics step, because that's something it's really difficult to change.
Okay.
Which trimmer you use, that's easy to change, but going back and redoing that experiment, that's a lot harder. So getting that right from the beginning is more important, I think.
Okay. On the topic of replicates, I read a paper that said ideally you have 12 replicates in any RNA-seq experiment, and that seems really difficult, especially when you're working with like hard to capture samples. So what are your thoughts on replicates?
The more the better.
Okay.
To be honest. I mean, I know 3 is like the magic number for a lot of people. I would say that can be okay, but then like if one of them fails, then you just have 2. So like over 5 I think is good, like 6 is great. I don't know where 12 exactly came from, but to give a sense, like in machine learning data sets, the like bare minimum for a lot of machine learning is like 50 samples.
Oh,
and for
okay.
deep learning, that's like hundreds of thousands of samples. Like 1,000 samples is not enough for deep learning.
Okay.
So there's always a conflict for biologists who are like, oh, I want to do deep learning. And you really don't have enough samples. You can't do deep learning on 10 samples.
Okay.
It doesn't work at all.
Okay.
But differential expression, RNA-seq is different.
Yeah. But I agree. More numbers is better. It's just daunting at times from like the biologist's perspective.
Oh yeah. It's always hard to get enough stuff, enough samples because every experiment's so hard and so time consuming.
Yeah.
So I want to see if we had any questions. Yeah. Because Ultra Lord is asking, how often do ticks come up as a conversation topic at your dinner table, and how do your family members and guests welcome such conversations? Is it true that lawyers are indeed blood-sucking parasites? Yeah, how do people react when you say you work on ticks?
I think often it's met with curiosity. I know when I told my mom that this is the lab I joined, she was like, ew, why can't you work on something cute? And then she had a lot of questions for me about like ticks and specifically what I was doing. So I'm really happy people come to me with their questions and then I can hopefully clear up misconceptions, because I think there are a lot of misconceptions about like how to properly remove a tick and if you're at risk for Lyme disease. I even get pictures of ticks people have encountered, so I can offer my services to identify them.
So do you get like random texts with a picture of a tick?
I
like,
do.
Is that like every day you get one?
No. Last time I got one I think was in the summer, but it was startling because it was a fully fed tick and I had to try and identify it. And they all look a little bit like beans when they're fully fed, so it's a little harder to tell what species this tick is. Yeah, I think largely it's met with amusement or curiosity. People aren't very... Yeah, I think people know that I kind of like the weird science, so this isn't a huge surprise.
What do you mean? Like, how did people know that you like weird science before? Like, what were you doing before that was kind of off the beaten path?
Well, I had really wanted... I guess it's not too weird in terms of like what goes on in microbiology, but I really wanted to work with the human gut microbiome, and so that means you're working with a lot of poop samples, and so I was aware of that, and it didn't discourage me. And I think I'm just always fond of like the unusual bacteria that exist. They're all weird and taking up every niche around us, so I'm pretty fascinated by them.
Well, and is that what got you to this lab?
Yeah, I definitely like bacteria. The ticks just came along.
So it's kind of funny that normally you would think of the bacteria coming along for the ride on the ticks, but here it's the other way around. Yeah, we had a talk recently at BioHub that was by Mike Eisen, and his this fungus that can live inside of Drosophila, of fruit flies, and basically like eat them from the inside and like take over their brain and make them these like zombie flies. And it's interesting because so he gives us talk and he starts off with saying, I'm like, firmly in belief that humans and other larger creatures are just here to serve microbes to get around and like reproduce, and like we are just like mere mortals, mere like servants to the microbes. I thought that was kind of funny. What do you think?
I think I can certainly feel that way, and sometimes when I think about like my food cravings, you wonder if it's how much of it is manipulated by like what's in your gut and what they're used to eating and what they would like to eat. So yeah, at a very high level, I do kind of wonder about that, but I'm not sure that the science is there to back up that we are really being manipulated by our gut.
Yeah, we don't fully know yet. Alright, another question. Can we make ticks glow in the dark so we can more easily find them in the dogs and family members? You can make GFP ticks, but then you have to use a fluorescent microscope. But you can use there's the new luminescent gene out, right?
In ticks?
Not in ticks, but I briefly saw this paper that they isolated the it wasn't luciferase from fireflies, but it was something else. It was another, maybe it was from algae, one of the bioluminescent genes, so that you don't have to use a fluorescent microscope. You can see the luminescence already without any additional tools that can just see. So that could be something that could be introduced to ticks, but I don't know how that would last in their population, because it seems like an evolutionary disadvantage if they're easily spotted by like birds in the dark.
Yeah, I agree with that. It's probably not going to be maintained if that was introduced in a while. But before we can get to that point, at the moment you can't introduce new genes to ticks. So like there's no CRISPR for tick species that we have. So we need to work on that first, and then we can begin trying to make our ticks more colorful.
Yeah, for sure. Actually, I wanted to show you this. So I had worked on, I was looking at some of the currently assembled genomes, because I was looking at the CNA's previous paper about the horizontal gene transfer.
Mm-hmm.
So I've been taught to be skeptical about horizontal gene transfer, so I wanted to look at the genome itself and look at the like splice sites and all that. And as I was trying to make that happen, then I ran into problems with, like, I couldn't search the genome. I could load it into my genome viewer IGV, but then the tool that you use to actually search the genome is not one that runs on your computer. It asks a program from a server called BLAT from the UCSC genome browser, and there's no, because I'm using a custom genome, it doesn't have that genome, I can't find it, and then I get stuck and I couldn't find it. Anyway, so I was trying to look at the, trying to find the assembly so far. So Zoltralord, I think part of the issue of why we can't introduce genes into ticks right now is we don't have a great genome assembly, so we don't really know where all the genes are in the tick genome, because it's not that well studied. It's not as high a priority as humans are, so we just don't have that data yet.
We're also pretty different. We're pretty evolutionarily separate, so I think that plays a big role. What we can do in human cells does not directly apply to what we can do in ticks or other species, but it's a good start.
Yeah, what kinds of experiments have you tried that are like classic mammalian cell experiments? Have you tried doing some of them that in ticks that just like totally failed?
I'm trying to do RNAi, so where you introduce double-stranded RNA and your RNA gets cut up, and it basically will match a mRNA sequence or transcript in the cell, and then that'll get degraded. So I've tried to do.
So it's like turning it off, right? So you're like, okay, turn this thing off. This is the thing I want to turn off.
Basically, I kind of think of it like it'll cut up the recipe for your particular protein, and I tried to do that with, I think, mixed success, but people have done it before, so I think it's just something that I need to tweak in my hands. But it's been published, so.
But the concept still works, but maybe like the specific concentrations of things, is that what you need to tweak?
I think that's a likely source, yeah.
Okay, cool. So yeah, so I was looking at some of these genomes, so because we got this JCDI ISE6 is the assembly from the J. Craig Venter Institute, the official ISE6 8.2.2. I think this is the one that they used PacBio on.
Oh, okay. Is this the embryonic?
I think
cells?
so.
Okay.
Yeah. I think this is, so they took a bunch of to catch people up, so they had a bunch of embryos from the female tick, collected them, smushed them all up, and made DNA from them. Oh, wait, no, I think this is actually the cell line.
ISC6 is the cell line?
Yeah. So ISC6, but is the cell line from embryos?
Yes.
Oh, okay. Got it. So yeah, so from ISC6 is the genome created from the cell line, and I think that's the current, like accepted draft, or just like the current.
I think it is I think the previously published genome was from the cell line.
Was it from the cell line? Okay.
Which I think is why we lack male-female specificity, because you don't have it that early on in development. But
Okay.
I would have to double check that as well.
Okay. So, so what we're looking at here is this assessment of the assembly using a tool called QUAST. We're looking at these two different assemblies. So one of them has a lot more pieces. This JCVI-ISC6 has about 19,000 pieces or contigs, and this other one has 10,000 contigs. But how does it make sense that this doesn't make sense to me. How is it possible that this one has 83,000 contigs greater than 0 base pairs, but 10,000 total contigs?
Yeah,
Shouldn't this number be the biggest one? How does this work? Oh, Zoltramore was asking me about males and females. Yeah, so maybe you can explain that again. There's a cool diagram for tick life stages we can look at. Is this a good one?
Sure, I'm not sure.
Hmm.
Oh, that's
Or this one? Which one do you like?
Well, I will say that one is maybe more relevant to what I do, because on the top we have Ixodes scapularis and then the Lone Star tick, which I showed earlier. But it's not that there aren't males or females, it's that until they reach adulthood, you can't tell them apart visually. And so when the people who were doing this original genome sequencing used embryonic cells, they had no way to know if they were a male embryo or a female embryo. I think
Yeah,
that's
that
the
makes
thing.
sense to me. Yeah, that makes sense to me. So we're saying that ticks have not evolved enough to the point where they have complex enough DNA. I think they still have complex DNA. It's just we can't tell by eye
Yes, and we don't have
that they're male or female.
enough genetic information to know. Oh yes, this is the male marker for ticks.
Do you mind holding this? I just have to charge my computer.
Okay. Yeah, so there definitely are males and females, and
Because there was a karyotype, right?
there has been a karyotype.
That showed that they're X and Y. Like we know at least that they're X and Y chromosome.
Yes. So they have males have an X and Y chromosome, and females have an XX, like humans. We just don't know exactly what is on each of those chromosomes.
I click in. Okay. Oh, cool. Perfect.
No problem. Ticks are pretty interesting. Once you start learning about them, you realize there's a lot left to be learned.
Yeah, and part of why I'm here with Fauna is because I'm actually working on this project with the group, working on some genome assembly, and where that's part of why we're working together, is that the genome still needs some work on the assembly part, and part of what's so hard is that like if you're thinking about genome assembly like a book, and you have we don't have technology yet to read the book from start to finish. You can only read like fragments of sentences at a time with one technology, and then that can let us to get like paragraph size segments, but you can't tell like which chapter the paragraph is in. And then there's other technology that lets you get like the order of the chapters, but it has tons of typos, so you don't have good you don't know exactly what words were there. So you have to do this whole mix of technology to be able to get it. So some of these genomes that we were looking at, where'd it go, was made with this technology that can make a lot of typos but lets you see the whole structure. And then the other one, this one was also made with another technology that doesn't go as long, and it has a little bit fewer typos. So that's the way we can think about it. Cool. Oh, sweet, you're sysadmin in North Carolina? Yeah, glad you're joining us. Biology is pretty sweet. There's a lot of really important problems that people are working on in biology, trying to apply computer science to it. So something that people are doing is using building genome assemblers. And yeah, glad you're enjoying this, Ultralord. It's always fun to chat with people. Yeah, sysadmin. That's also a thankless job. There's a lot of work you have to do to maintain it, and people only know you exist when things go wrong. Otherwise, people don't know you exist, which is kind of what you're trying to do, but then they find you and you're like, oh no, what happened now? Yeah. So what we're looking at over here is a comparison. Get away from the mail servers for 30 minutes? Yeah, I bet. I think that's gotta be tough. Let me get my water here. So what we're looking at is a couple different genome assemblies. So I was running this on, we actually kind of edited. I was kind of in the middle of editing it too. But we were looking at two different assemblies. One that we got from a collaborator that used technology that has a really long resolution, has low resolution, like it has a lot of typos in the genome sequence. Hi, that your fan. It has but you can see a lot. So it's from using a technology from a company called Dovetail Genomics. Dovetail. Yeah, so actually we can show you the paper, maybe. So here's the paper about that they use this technology. I think they have a picture at the end. Here we go. Let's just get some pictures. So what happens is that so DNA is not naked in the cell. It's covered in tons and tons of proteins. So the strand is the DNA, and then these circles are the proteins. And in real life, inside the cell, they're all tied together in a particular configuration. So what this technology does is take advantage of the fact that when things are close in proximity in the physical space, that they're often close in sequence space. And there are long tail interactions, long range interactions, and people do use this technology to study that as well. But there's always a really strong diagonal signal. Like if you have two chromosomes aligned to each other, or the same chromosome versus itself, you have a really strong diagonal. So it's really good to help you see what's the current, what's the shape of the chromosome, like what's the overall direction. So we have. So that's what these little technologies, or these little red things, are showing, is that what pieces are getting tied together, and then this technology adds on these little green pieces that are known, and then they do their genome annotation. So Ultralord has little questions about main arguments heard against evolution is don't see any half species walking around like cat, dog things. For this tick example you just talked about, they're too early in development. Is there an example like that concept?
So I think what I said was that if we think about evolution over time, humans and ticks have diverged a long time ago. So it's not that at one point there was like a half human, half tick ancestor. It's that there have been many species that grow more and more distant, and then they give rise to new species over time.
Yeah, so it's the one example I've heard is that it's less like you have someone who's like half one species and half another. It's more like we're like super distant cousins from ticks, and we have some like ancient grandparent that's like eons eons old, but we don't have but that ancestor gave rise to both of us. Let's see. Yeah. That your fan is saying better if a cat dog stays in cartoon, not real life. That's also probably true. Yeah, we don't have too many. I mean, one of the things that arguments I've heard about against evolution as well is that it is a theory, and it's a theory in the same way that gravity is a theory. Like, we can't turn it off. To really show that experimentally something is real, you turn it off and show it doesn't happen, and then turn it back on again and show that it does. And we can't do that with gravity. We can show that our rules are correct if the farther away from a big mass we go, from a big planet. And same with evolution, we can show that our theories hold up no matter how many experiments we do, but we can't turn it off. Like if we could, I don't know, like blindness versus like someone seeing. Like if you gave them sight, then they can see. You cover their eyes, they can't. Like you can't just turn it off and on.
I think some of that confusion may come from how theory is used in science versus how it's used like colloquially. Even sometimes I will say, "Oh, I have a theory about this situation," but really what I mean is I have a hypothesis about it, which is like I have an idea of how something might be working, and then you can test that hypothesis, and hopefully that hypothesis, well, not hopefully, that hypothesis will either support a larger theory or not. And so I think there's a little divide between like the language used formally in science and the language that's just used in general.
Yeah, I hadn't thought about that, because in science and in many technical fields, a theory that has a very specific meaning, like our sysadmin here probably has a very specific understanding of a container or a user, like that has a very specific meaning, versus someone who isn't in that field may not have those specific meanings coded to them yet. Yeah. My ears kind of hurt from having my sunglasses on, so I'm going to take them off. So this paper generated this genome, and that's one of the ones that we have, and then the other was this ISE6, I think, here we go. Yep. As you can see, I was looking at it recently. Here's the other paper with the genome. So this is draft genome for the Ixodia scapularis cell line from the...
This is actually very recent.
Yeah, I think this is one that that Michelle was looking at too.
Okay.
Because they used a different technology. I don't know if they have any nice pictures here. Well, this is decent. Showing different parts of the genome and what's haploid versus diploid. So I guess, how should we read this? How heterozygous it is, or like how different the genome is? Because if there's a lot of the genome that each of the chromosomes, both pairs of chromosomes are the same, is that something we should read as haploid versus heterozygous? So like if your two parents are really similar, right? So I think this makes sense in a cell line context. So like the parent cells of this cell are really similar, so their two pairs of chromosomes are also going to be really similar. So it could be that the genome looks haploid there, versus if the genomes are really different, then they could look more heterozygous or diploid. I think that's what we're seeing here. But they used a different technology called PacBio. sequencing. Yeah, the smart SMRT sequencing. So they do long read. Maybe we can find this little video to watch. So the way most sequencing works is that it has to, you do a sequencing by synthesis, that you basically, since the DNA is double-stranded, you can remove one strand, and starting from some known sequence, so you usually have to glue on stuff that you already know, and then you can add one letter at a time and see if it works. So here's which is ZMW. I think I missed what that stands for. Because here, PacBio is interesting because it has the one, I need to go back because I think I missed what ZMW means.
I think the ZMW is the well.
Zero more mode waveguide. Oh, okay, cool. So, so they have a tiny little light detection volume, so a tiny little piece that light can be found, and so it's illuminated from below. And this is the technology where it adds on one letter at a time, and it basically, that reaction creates light, right? That one, so they're attenuated light. I forget what attenuated means.
I would guess that means it's only a specific region of the spectrum.
Oh, right. Yeah, because it doesn't need to see like ultraviolet or like radio waves. Yeah, so you have some, they have a very specific molecular machine down at the bottom there that they add your DNA to, and then those little nucleotides that can get added, they're all labeled with a different color. So when they get incorporated, then a specific color flashes. So here, G is flashing, or yellow is flashing, and then they know they added yellow to indicate G, and so on. So as each molecule gets added, then it lights up, and you can tell which molecule it is. So that goes on for some time. This is nice because it has really high fidelity. It can go for a long time, but it is a little bit more error-prone than other technologies like Illumina sequencing, which has really low error rate but can only read for so long. Like 100 letters at a time versus this can do thousands at a time.
I think PacBio is interesting because I don't know if it's an official competition, but they seem to have this ongoing competition for who can get the longest read using this technology. And so I think last I read it was over 1 million. Is that right?
I know for Nanopore there was over 1 million, but Illumina recently bought PacBio.
Maybe I was thinking about Nanopore. Okay, but yeah, I didn't know. Someone recently told me about that purchase, and I was really surprised.
Yeah, Illumina is eating the world. So we have this genome report. So maybe I can actually rerun this, because I ran it earlier today. I was working on it earlier. But I was working on it earlier because I wanted to try. Yeah, so I tried it earlier, and maybe we can rerun it and use that other genome as the draft, or as the reference, because I think, what was it, tick, I should probably say species, so these Scapularis genome. There was I think this is it. Here's the paper. So genomic insights into the Ixodes scapularis tick vector of Lyme disease. So this is the existing draft. That's like the current accepted version, right? So let's take a look at where did they deposit their data?
I think you can get it from Vectorbase.
Okay, let's go there. Vectorbase.
A little frustrating to use, but
it is a source for people who work on vectors.
Huh. Is that just the kind of accepted one? Like, did you hear about it when you were in the lab here?
Yes. Yeah. But if you happen to be searching for mosquitoes or other things, you might find yourself coming across vector-based.
Okay, cool. I haven't really used it before, so I guess for the downloads, data files. So we want facilities scapularis and genome data. Genome data. Oh, okay, and this filtered it. Okay, cool. So what do we have here? Okay, it's got some of these files. Medium shifted genome. It's got genome filter. Do we have chromosomes for this?
As far as I know, we don't.
Okay. Vector-based ID to GenBank accession mappings. Well, that's actually useful. You can get some gene data too. What's this I3 one? Is that the previous version of the genome? ISCA W1? What are these genomes?
That might be.
Okay, it
I'm
looks
not
like
sure.
there's only these files anyway. So, repeat features, transposable elements. Alright, I see ISCA W1, ISCA W1, ISCA W1.6 gene set.
That may be the genome from the 2016 paper.
Yeah, I'm cool with that. Why don't I see this? What is just the like, genomic, okay, I think this is what we want. Genomic context sequences. And these are singleton sequences that weren't included. Oh, so this is a lot of extra stuff. Because look at the size of like this file. It's only like 450 megabytes. This is a gigabyte. So there's a gigabyte, like twice as many, just like random sequences. Okay, cool. I guess that's kind of interesting. Can we download this? Alright, and then I'll download it to my computer and then copy it to the cloud. But basically what I did was I ran this QuastLG program that evaluates genomes, so I can show you the parameters. So it said QuastLG quality assessment tool for large genome assemblies. So you give a bunch of files of contigs, and you can give a reference genome. So I didn't give a reference genome. I think if you, okay, yeah, file of genomic feature coordinates in the references. That will be good because I didn't run it with that. Because we have these draft genomes, and I think it would be good to get a sense of like how good these existing ones are, given the previous draft and all that. We can, let's see, gene set.
If there are big differences between the existing genome and like what you've been assembling, how is that read out? Like, how would you find out that there are problems or discrepancies?
Well, I found that there's differences based on what are the sequences that are mapping or not mapping to the genome. If there's a lot of sequences that are new, then again we'd have to be careful and make sure that these are real sequences, not bacterial contamination, and what I thought was kind of cool for this viewer is that you can look at, so you have this report that tells you like how many contigs you have. I still don't understand how this number is bigger. The like contigs bigger than 0 is more than the contigs period. Like I don't know if it's missing an order of magnitude or what. Like it doesn't make sense to me. But this one has like an order of magnitude largest contig. Although this one on the right has a longer total length of the genome. So this is a bunch of really small pieces, and this is fewer bigger pieces. So and I found there's this Icarus contig browser I thought was kind of nice to have it open here. But this shows you for the one assembly how big that contig is, and like so for the 25th, to these two different assemblies, you can see how different they are. Like this one on the bottom has a ton of little pieces, and this one the pieces are a lot bigger. You can also kind of scroll. Like this shows you your little views here so far. So the one on the bottom is bigger. So it's 3 gigabases, so it's like human genome size, which is correct from what I understand for the TIG. That is like between 2.5 to 3 gigabases is what I've seen. Whereas this top one doesn't have as many, isn't as long, but has a lot bigger pieces. So they have all these scaffolds. I thought in this paper they said they assembled it 2 chromosomes. Yeah, chromosome length scaffolds. Expanding on chromosomes. Haploid genome to 1 gigabases. The assembly consists of 3,609,495 scaffolds, representing 57% of the genome. Okay, where is this number coming from? 300,000? Is that? That must be from this previous data file, this one. So let's also make sure to download where the genes are. Base features. Sure. This is cool. I actually didn't know about this. This is really useful to have.
What is the GFF file format? I've come across that.
Oh yeah, it's one of many formats to tell you where stuff is in the genome.
Okay.
Yeah, all of these file formats are basically start here and end here, and there's stuff in between.
Okay.
And there's some that will let you link to other pieces. So like if you have genes that have the exons within them, so then you can specify, oh, here's these like sub-features of genes, and they all relate back to this one identifier. GFF is good for that, whereas BED has that. It is not quite as powerful as GFF in that respect. Like you just have to do a lot more work to get it to work. BED is really good when you have like from here to here there's stuff.
Okay.
Versus from here to here there's this like complex multi-part feature.
Okay, so GFF is a little bit more complete in some sense, or just more detailed.
Yeah, GFF is really good for gene annotations.
Okay.
Versus BED is good for like ChIP-seq or like other things where it's just from here to here, from like start and end. There's stuff versus genes can have really complex structures that aren't just start and stop. Like there's a bunch of other stuff happening between the start and end. Yeah, so let's take a look. So now I'm going to do some live coding here. I'm going to upload these to AWS. See, I think I have them. Let me go to downloads. This is hard to do with just one hand. Yeah, I gotta. Well, I should just really get a. I'm gonna hold this in my armpit, I guess. I should really get a stand. Oh, the lavalier mics are good too, but I have. Do this for now. Let me copy. What is it called? Series. Okay. ISCA ISCA 1. I'll call it that. Just capital I. We'll get that. Here we go. Context to genome, DNA, today's date, 1203. Let's see. This is KW1. I'll go with the same thing. I just want to. The dry run, it doesn't actually do it, just says, tells me what it's going to do. And sometimes I like doing that because I want to make sure I typed things right. And
So you would, if it was like if you type something incorrectly, you'd get your error message now instead of halfway through your command.
well, I get it says what it's about to do, so it will at least make sure that it's not totally messed up. Okay. Cool. So upload the context, and then we'll also upload the gene models because this program runs in the cloud, so we have to make sure that everything's up on Amazon, on AWS, the Amazon Web Services, before we actually run it.
What is a gene model?
Gene model tells you not just where, like, maybe you can like look at the genome browser for this. It's kind of a, maybe if they have a genome browser, that actually be kind of cool. For vector based, do you know if they do?
I don't. Well, I haven't come across it. If they do. That would be really nice.
Tools. Okay, actually, this is. Ah, cool. Oh, sweet. Yeah. Okay. Hey, are you gonna show me the alignment? Comparative. What does this mean? Ortholog 1-to-1. I thought it would show me. I want to see what it looks like in the genome. Okay, this is what. When I think of a genome browser, this is what I think of. I'm probably connected to mouse or something. Ooh, this is so, I don't even know what reason this is. Raw flow. Okay, let's go to mouse. So, a gene model is something that explains one of these lines. So each of line here is a version of a gene called an isoform. And so you have the start and end of the whole gene, where you also have the segments within. So like with the thicker parts are the exons that actually get expressed, and the very thin parts in between are the introns that get cut out, and the very ends, it looks like this little kind of hat, I think. The like thinner parts but aren't as thin as that line, those are the untranslated regions that don't get translated into protein. So the gene models are what show you where the transcript starts and stops, where the coding region starts, like where to start translating to protein. It shows you where the protein region ends, where the introns are, so I don't consider that as part of the protein. So this is what I expect when I see when I think of a genome browser. Like I want to see, here's the sequence, here's the gene aligned onto it. That's what I want to see here.
So the gene model info would be in that GFF file. Is that right? Yep. The gene model info would be in the GFF file.
Yeah. The gene models would be in the GFF file. I think GFF stands for Gene Feature Format.
Okay.
There's also GTF, which is similar.
Okay.
They can be basically interchangeable. GFF is slightly more modern,
okay.
but you can really use either. Most programs will accept either.
Okay.
Excuse me. Cool. Okay, yeah, so here's scaffolds, base features, peptides. Cool. So this is, I think this, yeah, so this is what we downloaded. Sweet. So now let's copy the, not the contigs, but the, what is it called? I have to hold this in my armpit again. No, I don't want to. What's it called? Base features? Yeah. Oops. We copy the whole file name. Otherwise it won't be what we think it is. So I then think in this we can, I think I'm just going to specify. Alright, Soltero is back after eating tacos. Cool. I'm just going to hard code in the genome, reference genome for now. I'm going to change it later. But since this is the only genome I'm working on, that's what I'll do. How are your tacos? Right now what we're looking at, we're adding in some the current reference genome to our genome assembly assessment program. So we were assessing some existing genome annotations and realized that we can add the previous reference genome to compare to, so we're adding that up. I'm going to call this ID ISCA W1. W1 and gene annotations for reference genome. Okay, first let's see if this will run, which it probably won't, because they never do the first time. So if I run the doc, oh, actually there's no syntax errors. That's good. Oh, but I think I didn't add the gene stuff to here, so I should just say -r, what is it? -r for the reference genome file. Reference file. We have to do some kind of funny things to convert these into files. Reference. Alright, Salterlord had some get Taco Bell, but DoorDash delivers Taco Bell now. That's pretty amazing.
I do love Taco Bell. I live 2 blocks from a Taco Bell. I don't eat it that often, but it's there.
Wow, nice. Homemade tacos are pretty amazing, though.
Yes.
Do you actually like holding this?
Oh yeah, of course. So it looks like you are pretty great at commenting your code.
I definitely try, because if I didn't, then I would never know what I did yesterday. Okay, let's do -- features, and that's the next thing. So we have to add reference genome as a file input. We also need to add the reference genes. Reference features, I guess. I'll put that here. Right. It takes a little getting used to this programming language. This what's this called? Workflow language called Reflow, but now that I've gotten used to it, I really, really like it.
And did you just start using it for the assembly?
I had been using it a little bit, but I say for this project and one of my other projects, it really got me. I had to use it. There was no getting around it, so it got me into it a lot more. Yeah. Yes, you do eat Taco Bell. If it's that close by, then there's no way that it gets passed up. You're publicly denying Taco Bell right now.
I do eat it, just not as often as I could eat it.
Yeah. Okay. Also, this program's a little funny in that I think I have to unzip stuff. Oh, hello from hello from Russia. That is their username is a bunch of bad words in Russian.
Oh.
So we won't say what they are.
Okay.
But we need to make their reference IDs. So one thing that's kind of annoying about this program is that it completely abstracts away file names.
Okay.
So whatever you put in as like the first argument gets turned into /arg/0/0.
Okay.
So it doesn't have a .fasta or .gtf or whatever file txt file ending anymore. And a lot of bioinformatics programs depend on the file endings, so you kind of have to hack that in.
Okay.
Yeah, you have to hack that in. All right, we'll accept that answer because we're scientists. That's right. Real scientists. Real science right here, right now. All right, so I want to make the reference dir, so I have to make this to make like a reference ID. Live coding. This is always fun because you get to see all the mistakes I make. Okay, so we're gonna do reference ID plus .fasta. Oh, really? The audio coming from my laptop mic? Uh-oh. That is not what we want. I wonder if this connections are kind of loose. Just, is that better? Just unplug everything and replug it back in. Is that any better? Probably the same. Hopefully it's not too echoey in here though. Let's see. To reference genome, and then I misspelled that, reference plus, what kind of file do we want this to be? I think GFF.
So then it'll print out what was the original file type because of what you're doing now?
It'll make a pretend file
Okay.
that has the right name to trick this program into thinking these are the right file types, because a lot of programs depend on the names.
Okay.
Yeah. Alright, sorry for the nickname. It's okay. Just Russians know. That's fine. Alright. So I think we can make this directory, and then here we'll say, instead of reference genome, we'll do reference dir / and the reference dir is going to contain this reference ID because we made it reference ID .fasta file, and then reference dir / reference ID .gff3. I think actually I've been using this extension variable. I think that will be better because it is actually zipped. It's like not the raw file, it's zipped, it's compressed. So let's do that. Alright, so I've written a bunch of stuff and probably something is wrong. I also need to unzip this GFF3 file because I don't think this program will accept it. Yeah, so references in GFF, bed, NCBI, or TXT. I don't know if it has to be. Let's check Quast, GFF, gzipped. I searched for Quast gzipped earlier. Files may be compressed with gzip. Sequences. Okay. Cool. So all of these are allowed to be gzipped. What about the GFFs though? Not compress this. But it only said about the FASTA. Yeah, I don't want to risk it, so I'm gonna unzip it. Okay. Sorry, this is so boring, but this is what science is when you're actually doing it live.
It's very helpful.
Okay, so I had, you know what, to unzip it. Parallel unzip, but I forget how you run it. I'm just gonna Google it. PIGZ unzip decompress -DC for decompression. Okay, cool. I think we just had a D here, and now it's decompressing. Unzip, so we'll call this unzipped. And this will be the input file name. Cool. So we'll do this GFF is utils dot, oops, what was it called? Parallel unzip. Reference features, and we'll do 8 threads. And then, so now we want to do, let's close GFF. So we add the IDs. The next thing is the reference ID, reference genome, reference GFF. Okay, maybe this will work. Cool, okay, well it doesn't have any syntax errors, so that's good. We'll add Quast and utils. We'll add that a message that we added reference genome for Quast comparison. And git push.
Do you think this comparison will go any faster now that you have a reference?
No, it'll go slower for sure.
okay.
Because it has to, because before it was just looking at the number of contigs and all that, and now it's going to do an alignment.
Oh, okay. Yes, I know how long that can take.
Yeah. You okay?
Yes.
You have your phone there?
Yeah.
Alright, so we can try and. Alright. Sosu. I guess I can call you. It's like the. I'll imagine it's like the pacifier that kids have. That's one of the nicknames for it. But how's Russia? Where are you in Russia? Let's see if we can get back to my super machine where I'm actually going to run this stuff. I think I have to VPN first.
So you'll be running stuff on AWS or the BioHub machine?
So it's really complicated. So that's a good question. Oh, St. Petersburg, cool. I've never been. I heard it's very beautiful. I have to remember to type my password actually correctly. Well, I don't think I did that right. Oh. Hello, Paul person. What are we doing here? That's a good question. We are assessing some genome assemblies. So we're working on the tick genome Okay, make sure I do this right. Copy password. Okay. Olga.bot. So we're about to run a genome comparison or genome assembly evaluation tool. Let me close some of these other tabs so I don't need these. Okay, let's go to our super machine. So I'm going to log into this super machine on called Unda on BioHub. So it's like, I don't know, half a mile away, 40 some amount of feet away. But it's going to launch things that are on launch jobs on AWS.
Oh, okay.
So the orchestrator of the project of the system is going to be here, or sorry, going to be at BioHub, but then it will launch jobs in the cloud.
Okay. So it's launching that many jobs that it can't do it locally?
No, I could run this at BioHub, but I don't know. I just chose not to.
Okay.
I guess. Am I studying now? No, I'm actually doing real work. This is my job. I'm one of the few people on Twitch that actually gets to show their real work, which I think is pretty cool. Because the work I work on is all open science, open data, so you don't have to worry about proprietary secrets getting stolen or whatever. It's just science.
I saw that when you tweeted about this today, you referenced the foundation. What was the foundation?
Oh, the Knowledge Fellowship?
Oh, fellowship. Okay.
Yeah, so it's a group of people at on Twitch who are educational streamers. So there's quite a few actually that are streaming educational content. So there's someone called Dr. WD40 who is also a biology person, biology professor who'll host office hours on Twitch, and they do a lot of people do both gaming, which is what Twitch is normally known for, and educational content. So they'll some of the times stream them playing games, other times, like yesterday I saw someone streaming, they were looking at the week's science news and talking about it. I saw someone else that does like all of the New York Times crosswords like in a row for the week for like 4 to 6 hours. So I think that's pretty cool.
They get progressively harder, right?
Yeah. Well, they get harder. The thing is like Thursdays, because I like crosswords, so Thursdays are like pretty hard, and like Saturdays are actually some of the hardest. Like Thursday to Saturday are some of the hardest, but Sundays are just really big.
Oh.
So Saturday is the hardest, but Sunday is like a Thursday difficulty, but really big.
Okay.
Yeah. But Monday, Tuesday, like I can usually finish a Monday, Tuesday without looking anything up, but Wednesdays I have to look at stuff up. I'm not that great yet. But it's kind of cool to see it. But yeah, check it out. The this little icon from Paul Person, that's the Knowledge Fellowship little icon. So it's kind of, you can go to it. There's a, you go to knowledge, oops, if I can spell, knowledge fellowship, Twitch. Yeah, people like post their schedules and join the fellowship. So this is someone in Florida doing. There's a Dr. WD40 doing it. Rocket Sage is a geologist who I also check out sometimes, and so it's a nice little community because everyone's like really into science. So it's nice and positive and all that.
Great. I had no idea until today.
Yeah, it's interesting. Actually, before I started streaming, I hadn't really watched Twitch much even. I just kind of knew it existed and had thought about doing live streaming of coding because I thought that would be fun. But I didn't know how many other people were really doing it, or that there was this whole community for it.
Yeah.
Yeah, so here's... I think there's probably a link for the Knowledge Fellowship in here somewhere to join the group. Just hosting someone else now. Yeah, I think if I right-click this, I can get the link. Oh, I think that's just more info about the Knowledge Fellowship. But, yeah. Yeah, so I can actually, from Susu, I can show you the project that we're working on is, so it's here, so it's called Tick Genome from Suzy Biohub, and the goal is to assemble and annotate the tick genome. So I'm going to add this pull request for my pushed branch. So I'm going to call this add, I'm going to say work in progress, WIP for work in progress, add WAST, all caps, genome, assembly evaluator. Oh, I actually typed that right. Cool. So yeah, this is the project where I don't think it's structured like a normal software project with tests and all that. Oh, okay. Am I teaching her? Kind of. We're collaborating. So I learn from Fauna and she learns from me. I think that's the goal.
Yeah.
So we teach each other. Oh, here we go. KnowledgeFellowship.com. Cool. So yeah, they have the official Twitch channel, it's a little Twitch icon, and then Discord, which is kind of like Slack, basically. It's like...
Oh, I've heard of Discord.
Yeah. And so you can join that. It kind of looks like Slack, to my eyes at least. But it's a little discussion board. People can post like when they're online and discuss stuff about science and whatnot. So it's cool.
Can we talk a little bit about GitHub? Because as a novice bioinformaticist, GitHub seems a little bit intimidating, but I think it can also, like, my understanding is it can be really powerful.
Yeah, for sure. Yeah.
Like, how should I get started, I guess?
In GitHub? Yeah. I think for example with this project, one way that you can get started is by making issues or commenting on pull requests. So what this view is like all the files that you'd normally see when you look at the code, like in the file, like in Finder or file, whatever file browser you have for your computer. And what's nice about Git specifically is that it has a history of all the changes ever made to all the files. So we can go, like, we can go back in time. We can press this really fun button called Blame, and we can look at like when I added different changes, and even like revert and like see, okay, like what happened here, like what was wrong here, like what did I add at this point, what was my message. So when I enter a commit message, this is where it gets saved.
Okay.
And it's a really nice way to keep track of your code. Otherwise, if I was like changing code and changing one line, I need to go back to some change, or like right now I have these, I really have like these like 6 things in flight, these like 6 like changes I'm doing at the same time. And because of Git, it's really easy for me to change from one version of that to the other without worrying that they're conflicting.
Okay.
So it's a nice way that I can work on like one part of the code or a different part of the code without being like, oh no, I'm changing the same thing. Like if I change this in this version, it's going to mess up this other thing. So that way I can keep it all really separate.
Okay, so you can kind of branch away and maybe tinker with one part.
Yeah, yeah, that's exactly actually what it's called. It's called branching.
Okay.
So yeah.
Cool.
How long am I in development? I've been working on this project since, I guess we can look. When was my first commit? I feel like it was sometime in the spring. I think it's in Insights. Traffic? Contributors? But I work very slowly because this is my first genome assembly, so it's taken me a really long time to learn everything that you have to do. So it's been a while to just understand everything else you have to do. Plus, this workflow language is new to me, and it's like super like mess with my head at the beginning, and now I get it. But I had to like change the way I think about things. So I would say so. Yeah. So first, since July 1st, I started until now. So just under 5 months, I guess. Not too long.
I think in general, science takes longer than you expect.
Oh yeah.
And then, yeah, my experience with sequencing and sequencing data is that the learning curve is a little steep, and so it is taking longer than I would have liked or anticipated. But stuff's happening.
Yeah. Yeah, one of the things that I kind of like about GitHub is that we can use these projects and these little like project boards. I don't know if you've seen stuff like this before, but it's like a different. It's called a Kanban board, but I think it was pioneered by people at Toyota, but to as a sense of like what. Like what's to do in progress and done, but like way to keep track of your project tasks and stuff. Because I was thinking, like we could have a project for the like transcriptome stuff too, right? Like we could add. We can even do that now.
That'd be helpful.
Oh yay, Jesse Skinner learning about GitHub features. Sweet. Yeah, so we can say, it's not Scapularis, it's a bunch of different species.
I think right now I'm focusing on Scapularis, but yeah, we do have other species transcriptomes.
Okay, we'll just call it transcriptome. Transcriptome. Oops, Не можно писать на русском. Можно? You can write in Russian. I can read it. We can do, I think I did the automated Kanban. Here we go. With reviews, no. This one's kind of nice because as you, so for example, if you have something that's in the, that's a project that's a pull request, so when something is when something is a pull request, here, I think I can show you. Wait. How do I add? Close this. Archive. Oh, I think I want to go here. Ты работаешь не в России? Yeah. Like they're asking, do I work in not Russia? I work in America. So what we can do is we can add this to Забавный акцент просто. Oh, I have a funny Russian accent. Yeah. Well, because I'm American. I can't help it. Sometimes I try to have less of a American accent, but that's just how I am. So I can add this project, this pull request, this genome assembly evaluator. So what a pull request is, the idea behind it is that there's the main branch, or like the main trunk of the project. In other projects, it's actually called a trunk, or in other languages, it's called a trunk. But you have all these branches that you're working on, and when you want to put them back in, the pull request is yours saying, hey, I have this cool code. Can you? I request that you pull my code into the main branch. And that main branch is called master in this case. So I have this pull request. It's a little contrived because I'm doing a pull request to myself, to my own code. But it's just good practice that you don't just like keep changing the main branch, and then like it's hard to keep track of what's, like I said, if you change one thing that affects something else, that like when that happened, it just gets really hard to track that. So you want to make sure that every time you add a thing, it's like a complete thing and not a fragment.
Okay.
So that's what the idea of the pull requests are. So did I learn Russian in university? I learned it mostly at home, speaking with my family, but I did take Russian grammar classes in college because my grammar is really bad and I speak like a 6-year-old. So I tried to learn grammar so I could speak at least like an 8-year-old. But I'm somewhere in elementary school with my Russian.
I speak a little bit of Spanish, and I'm also in elementary school, and often surpassed by students in elementary school. So.
Yeah, it's kind of embarrassing sometimes.
Yeah. What is that face?
У тебя очень хороший русский, если ты его учила. I have good Russian if I learned it, taught it in school. My accent is alright, but yeah, my grammar is like, yeah. I wrote happy birthday card to my grandfather for his birthday, and was like, oh, I surely can get this right. So I just wrote it without checking, and then I gave it, and then I was like, oh shoot, I think I got it wrong. So I asked my grandma, and I was like, oh, did I get it wrong? She's like, yeah, you should have done it this way. Oh shoot. So I went and corrected it in the card. I can't believe I got just like happy birthday wrong. Like I should have been able to spell that correctly or like get the grammar right for that.
You probably learned.
Yeah, no, I learned. It was good. But I also appreciate it when people tell me correct my mistakes because otherwise I don't notice them and don't know I'm doing it. So my grandparents are like the ones that would correct me, but my parents don't correct me as much. So I have to remind them to correct me. Yeah, and this is a little like thumbs up face for. So here's this genome assembly evaluator thing we're adding. One thing that's cool is that you can see what changes you've added. So in the green here is showing what has been added. So here I added this few lines to what's called a make file. These are really useful files that you can use to basically summarize a bunch of commands into one smaller command. So here I'm running, I have this little like test quast command that I can say make space test quast and it will run like this whole thing. So I don't have to remember like with the genome locations every time or the name of the things and all that.
So that's your dry run.
This is the actual run. This will actually run. I think there might be a dry run option, but I haven't used it. There's the dry run option is the doc, reflow space doc.
Okay.
That will just check if the syntax is right.
I see. Okay.
Yeah. One of the things that's kind of nice, that's nice about this language, but can be frustrating for people like me when you're first starting with it, is that it's very particular about types. So if you say something is a file, and then you gave something should be a file, and then you gave it a directory, it's like, you said this was going to be a file, why are you giving me a directory? Like, I don't know what to do with this. And you're like, okay, fine, I'll give you the one file in that directory, but like, can you stop yelling at me? I just want you to do the thing I told you to do.
Yeah.
So computers are like that sometimes. So we got the thing running. Okay, cool. So we have a nice new error. So it started running some stuff, and it found the, oh, awesome, check this out. Look at this typo. So I did reference dir contains iska w1.fasta.gz, and it has this unique identifier, this SHA, and this like long ass string. And then it has iska w1 gff3 no dot, but I told it had a dot. So now I have to go back and add that one dot. You see?
Oh.
forgot
Yes.
the dot.
Yes.
Yay, bioinformatics. Okay. So now we're gonna commit that code, add it again. We're gonna say added dot.
And so when you, oh, I see. So you are running git add, so then it'll,
So it adds the yeah.
okay,
Go ahead.
it'll automatically like deposit that in your GitHub account. Is that the right way to think of it?
Yeah. So the way it does is I'm adding the one file, and then you can add many files at a time for a set of changes, and then you always have to have a message, a commit message for them, and then I'm pushing it to GitHub, and then I'm gonna pull it back down, not from my laptop but from this other machine. So now I'm on this other computer, and so if I pull it, it should pull a bunch of stuff, and now it's gonna run. So I said make test quas. So if git pull and means do the previous thing, do the next thing if the previous thing was successful. So if it didn't fail, if the previous thing didn't fail or like error out, then do the next thing. So the next thing is say make test quas. So this first part is all the git stuff, doing the git pull, and now here is where it's actually running that make test quas. So this make test quas is running all of this file.
And it,
All right. Yeah.
Oh, it actually tells you how expensive it is to run
Yeah.
on AWS?
Yeah. For this case, it does. For Reflow, it does. Not all of them tell you that.
Okay.
Yeah. Which is kind of funny. Right, we have хочу уйти с университета и заниматься... Wait. That's a long-ass Russian word. Okay. Заняться программированием сам. А какое твое мнение насчет высшего образования? Обязательно ли он программист и как с самого начала программировать? Okay, so the question is, I'm thinking about leaving university and learning to code on my own. What do you think about that? Is higher education necessary for being a programmer? And what is and how do you actually start programming? Yeah. Yeah. Oh, thanks for telling me about the lag. Yeah, I think the Wi-Fi here is not as great as is at my house. Is okay. Well, at BioHub it's really great, except when it cuts out. But overall, hopefully the lag is all right. Maybe I should close some programs so my computer's less tired, because that always affects it too. So you can see I have a lot of stuff open. Ooh. But when it comes to programming and higher education, I would say that having a computer science degree makes it... Oh, Jesse says you can set an offset in the audio settings. Interesting. Okay, that's something I'll have to experiment another time. But I would say for getting a programming job, it's going to be a lot easier to get a job with a computer science degree than without, because I think you would have to have a pretty compelling portfolio of like websites or recommendations from people to get in. To start programming, I mean, I always say the best way to learn is to do a project you care about. Like, I know people who are like doing RNA-seq and they're like playing around with R and Python and they're like doing some tutorials, but all they've done is like existing data or some like existing data that's out there, or like a classic data set, like this IRIS data set that's used, this classic data set used in machine learning. But it's really hard to motivate yourself.
Yes.
To try some random algorithm on this data set that doesn't matter to you. So I would always say get tutorials are a great way to start, but like think about how you can tweak that tutorial or change it to be relevant to you. So, like working on a project that solves a problem that's relevant for you. Like, I don't know, the other day my boyfriend and I were making food, and we had like 6 recipes we needed to make, and we were like, oh my god, like if only there was like a Gantt chart that we could use to see what's the optimal flow for all these recipes. So if you make a Gantt chart for me for all my recipes, that would be sweet. But yeah, I think something that solves a problem for you, because ultimately that's what programming is about, is like solving a problem that you have, like whether it's finding what genes are different or like finding like clothes to wear in the morning or food to eat. Like that's all I see it as, is like solving that problem.
I agree that you should find something you are not going to easily give up on, so something you care about. And I also think in terms of staying in school or going to school in the first place, I think, or my understanding is it's possible to get a job without a degree, but you might find yourself being unable to advance in your company or in your profession without that degree. So I think that's one more advantage to going to school and graduating.
Yeah, for sure. I know a lot of people end up getting their PhDs because they're like in biology at least, that they're like technicians for a long time, and they are really doing the work that's like equivalent to a PhD, but like because they don't have that degree, they can't make it to the next step that they want to be at, which is too bad.
Yeah.
But that's part of the system here. So Paul Person suggesting get a GitHub and jump on with some tutorials. Easy way to start up and see if you like it. Actually, that's actually a good point about GitHub. Something I want to point out is if you have, say you're doing machine learning, and you're really liking your machine learning tutorials and you think they're pretty awesome. One great way to get more into the community is to contribute to the code. So like that's one way that you can definitely get noticed and definitely get appreciated, is if you go to a mature project like scikit-learn. A lot of them will have labels like this where they'll say, like, for example, good first issue, or documentation errors. Like maybe you were reading the documentation and you noticed something was wrong. Like it can be really easy to change that. Oh, thanks, Paul Person. So I would say definitely check out open source projects and contribute there, because that's a really great way to, one, get practice actually contributing to code and like dealing with standards that you don't have when you're in a class necessarily, of like, you know, because it's part of this project, it has to have all the lines can't be longer than 80 characters, or they have a particular style guide that they follow, like all their doc strings have to be a particular format. And, every project is different, so it's good to get used to the different styles and used to having rules like that you have to follow. So I find that to be really useful. So yeah, I'd say definitely check out like good first issues or other kinds of resources. Any mature project will have that. So any library that you would normally import that is like famous is going to have a pretty mature code base, and that is something that you can contribute to. Yeah, so I think it's actually time. I think that time actually flew by pretty quickly for me. Maybe partly because we had some technical issues and had to start late. But yeah, thank you everyone for joining and asking questions. Really appreciate everyone. And thank you especially to Fauna for donating time and talking us ticks with us and showing us some live ones. We didn't see any dead ones today.
Not that I know of. They could have been in there.
Okay. Okay. All right. Well, thank you, everyone.